Hi everybody, Dr. Ahles here. In this short video, we're going to talk about some of the key scientific discoveries that have helped us to understand DNA. When we talk about understanding DNA, it's important for us to think about a couple of dates. We talked about Mendel and how he helped us to map out the way that traits were passed along from parents into offspring. Remember though, Mendel's work with pea plants happened in the 1800s, and back then we hadn't even seen chromosomes yet. He had no idea what was being passed from one generation to the next. That kind of work happened in the late 1800s or even into the 1900s, when we start to see some of these important experiments that we're going to talk about today. So the first important experiment that we're going to explore is the Griffith Experiment. The Griffith Experiment helped us to understand that living cells, not even just parents to offspring, but living cells can actually transfer their genetic information between each other. The next important experiment is the Avery Experiment. The Avery Experiment built on the Griffith Experiment. The Avery Experiment showed us that what it was that was being transferred from one cell to another was the DNA. The final thing we're going to discuss is how we figured out the structure of DNA, the double helix, the location of sugars versus bases. This was work done by three scientists, Rosalind Franklin, James Watson, and Frederick Crick. So let's start with Frederick Griffith's experiment. Frederick Griffith was a microbiologist who worked with a type of bacterium called Streptococcus pneumoniae. And if you look at our picture over here, you can see a nice electron microscopy version of this bacterium. Streptococcus pneumoniae comes in two main types, two main strains. One of them is called the R strain. And the R strain of Streptococcus pneumoniae is harmless. It's not going to cause an infection. You can have those cells inside of you, no problem. But the S strain of this bacterium is deadly, meaning mice that are injected with the S strain will die because of this bacterium. Griffith was interested in seeing what would happen if he injected live forms and dead forms of both of these kinds of bacteria into mice to see if they would develop this illness in their lungs. What he was studying, though he couldn't quite put his hand on it, was this idea of transformation. Bacteria have this unique ability to share information with each other. Griffith didn't know what it was that they were sharing, but he found that a harmless bacterium could be transformed into a deadly bacterium because the deadly bacterium shared something with that harmless bacterium. So let's look at the setup of Griffith's experiment. First, to make sure he could prove his cells were viable and were working, he started by injecting mice with either the S form of living bacteria, remember this is the deadly form, or the R form, which was the harmless form. Notice that mice who were injected with the living S cells, the deadly bacterium, died from that bacterial infection, while mice who were injected with living cells of the R strain, the harmless one, had no problems. They remained healthy. So Griffith thought, what if I kill the S cells? Would the dead S cells by themselves lead to an infection in the mouse? When he injected those dead cells into the mouse, remember this is the harmful strain, when those bacteria were dead, that mouse was totally fine. It was safe and healthy. So then Griffith took dead S cells, the deadly cells, and injected them into mice at the same time as the harmless cells that were alive. He watched, and to his amazement, that mouse died. But what was interesting about that mouse dying was that when Griffith looked inside of the mouse, he actually found living versions of the S cells. Remember, the S cells that he injected into the mouse were actually dead. he had only put living R cells into that mouse. So what Griffith discovered was that his bacteria had transformed. They had gone from being live, harmless cells into live, deadly cells. The process of transformation had occurred. Griffith showed us that transformation can happen, that a dead bacterium can be used to change a living bacterium. Avery picked up where Oswald ended. He was again using the R strain and the S strain of Streptococcus pneumoniae. What Avery wanted to see was if he could figure out exactly which part of my deadly bacterium is what was leading to an infection in the mice, or what was being transferred from one bacterium to another. So the way that Avery set up his experiment is he again used heat-killed S strain, the deadly strain that was dead. He added enzymes one by one to break down individual components of that deadly bacterium. So he used an enzyme called proteinase. Proteinase breaks down all of the proteins that a bacterium had made. He used RNase. RNase broke down all of its RNA genetic information. And he used DNase to break down the DNA. Let's look at his results. First, we had the heat-treated, killed bacteria that we added the enzyme proteinase to. Proteinase breaks down the proteins. He took this mixture that now had RNA and DNA in it, and he gave it to bacteria that were alive, the harmless bacteria that were alive. He checked out what he found with the bacteria after that, and he found some of the living S strain, meaning whatever it was that transferred from one cell to another, it wasn't proteins. We still had some of those live S cells that he discovered. Next, he took the same heat-killed bacteria and treated them with RNase. RNase breaks down all of the RNA that the bacteria had inside its cell. He took this mixture, the one that didn't have RNA anymore, mixed it with the harmless bacteria, and looked at what he got. Again, he got bacteria that were that deadly S strain. Whatever it is that's being transferred, it's not RNA, because we still see our bacteria being transformed. Finally, he took that same heat-killed mixture of the dead bacteria and he added the enzyme DNase. DNase broke down all of the DNA, leaving behind RNA and proteins. When he added this mixture to his harmless bacteria, let it mix together, he only ended up with harmless bacteria. Because this was the only test where our bacterial cells did not transform, this meant that the enzyme deactivated what the cells needed to transform. So because the bacteria did not transform when the DNA was missing, this meant that the DNA is what led to their changes. So let's recap. Number 1, Griffith's experiment. Griffith told us that a dead bacterium, the deadly form, can change a harmless bacteria into a deadly bacteria. That was Griffith. Avery said the way that deadly bacteria is transforming our harmless bacteria is DNA. The final set of experiments I want you to be familiar with are the work of Rosalind Franklin and Watson and Crick. Rosalind Franklin worked with a man named Maurice Wilkins, and together the two of them took a series of X-ray images of DNA up close. So this is the kind of image that Rosalind Franklin was able to develop of the structure of DNA. The way this image works is areas that are darker are places where those X-rays bounced off of the structure of the DNA molecule itself. By using this X-ray structure, we could see that DNA was in a helix, or kind of in a back and forth twirling pattern, a helix. That was the work of Franklin, taking X-rays to show us the structure of DNA. Using those X-ray images, James Watson and Francis Crick were then able to figure out the overall 3D shape of DNA. Their work was a little bit more hands-on, or a little bit bigger scale. What we're looking at here is a picture of their first model of what DNA actually looked like, or their first correct model, we should say. Because when Watson and Crick first started building models of DNA, they started by putting the sugars, the deoxyribose sugar, in the middle of the double helix, and tried to put the nitrogenous bases on the outside. But when they put that sugar in the middle of the helix, it ended up building a shape that didn't match up with Rosalind Franklin's images. So a big breakthrough came when they put those sugars on the outside of the helix. Once they put the sugars on the outside, the measurements from the X-rays matched, and they were able to figure out that DNA is this double helix form. Watson and Crick also were the ones who helped us to figure out which bases paired with each other by, again, using measurements that they took from Rosalind Franklin's images. So as we wrap up this first chapter on DNA, remember that our pathway to discovering it involved bacteria, it involved mice, it involved X-rays, and as we see in this picture, a whole lot of wire in a silly looking model.